Thank you so much, Peter. It's an absolute honor to be here with you all, to share with you our vision of how we can build AI systems that can enable and accelerate new biological discovery towards a vision of precision health. Our work is fundamentally motivated by the fact that human health and human life is a marvel. On one hand, incredibly robust, and on the other hand, incredibly delicate. And to me, the most amazing thing about all this is that each and every one of us is unique. We all have different bodies, different genetic makeups, a different history of experiences that makes you, you, and me, me. And yet, somehow, it all seems to work in concert. And yet, shockingly, in medicine today, the fact is that these nuanced differences, these richnesses and complexities, are largely not taken into account. To preserve our health in a way that's truly personalized, we don't just need a better way to detect and treat disease or discover new drugs. I'd argue that we need a whole new way to think about how we unlock new biological discoveries and learn those patterns that differentiate us all and deploy them forward through the use of powerful tools like AI. Now, it's no secret that today we're experiencing a revolution with a new generation of AI systems trained on words and text, the language of us as humans. But yet, there is tremendous opportunity to unlock and learn the language of biology because of the fact that biology, through the hand of evolution, naturally stores an incredible scale, richness, and complexity of data within each and every one of us. Our vision at Microsoft Research is to leverage this opportunity to now develop new AI towards the vision of personalizing medicine, to help us discover the mechanisms of disease better, to design new treatments, and ultimately deploy these systems into the real world in a way that's reliable and safe. Let me start by showing you why this matters and a concrete example of why we should care. Let's consider cancer as a use case. In the United States, nearly 40% of the population will develop cancer in their lifetime. And while we've known for a while that cancer is driven fundamentally by changes in the biology of our cells, we yet don't understand how to condition the therapies based on those changes to deliver the right treatment to the right patient at the right time. Indeed, for fewer than 40% of cancer patients today, there exists what we call targeted therapies that are therapies specific to changes in their cells. But what's even more shocking is of those 40%, less than 5% of patients even respond effectively to today's treatments. So clearly there's a problem, and clearly we can do better. But why are we failing? The answer to that is that because, by and large, cancer is treated in this ad hoc, reductionist way, where that richness and complexity of the individual is lost, because they're viewed as rather an instance in a population, given a treatment that works on average over large-scale population-wide studies. And so I've been speaking a lot about this richness, this complexity, this scale of biological data. What exactly do I mean by this, and how can we make this concrete? I'd like you to consider now, suppose we have one cancer patient, and we take a biopsy from one tumor from that patient. At just this level, thinking about the scales of biological resolution, from the level of DNA mutations to the transcriptome, to proteins, to the interactions of cells in their neighborhoods and how they communicate with each other, we can generate nearly 50 million individual data points from just this single tumor biopsy using our measurement and experimental techniques that we have in the lab today. Now, let's pause for a second and consider this number. Right, when I look at this as an AI researcher and as a computer scientist, I see tremendous opportunity. And if I look at this number as a biologist, I see power that comes at the hand of this richness and complexity. What's beautiful is we can put those together by using this ability to directly measure biology at its natural scale, the nanoscale, as it's occurring in real time. This gives us the opportunity to unlock the development of new AI. And today I'm very excited to share with you 2 concrete ways in which we at Microsoft Research have brought this to life. First, in collaboration and in close partnership with the Broad Institute of MIT and Harvard, we're collaborating to create a new vision for precision oncology, closing the loop on this vision that we've been talking about by putting AI front and center, such that we can directly take these molecular measurements from the patient level, train and build AI systems that can now produce recommendations about what experiments are best to test for that patient in a personalized way. We can use this as a flywheel to generate the new data that we produce in the lab, to then iterate and improve our AI model, and ultimately recommend or design new treatments that are specific and tailored to the needs and biology of that person. We can dive deeper on one of the components of this pipeline and ask, what does it take to actually design new molecular therapies, new treatments that have functional ability? And to this end, we've built and are building powerful new generative AI systems to design new molecular therapies like proteins. By learning the language of protein function, learning from large-scale, evolutionary-scale data sets of 50 million unique protein sequences, we've trained a new generative AI model that we call EvoDiff that can now produce brand new instances of proteins that have never been seen before in nature, with the goal of expanding the functional capabilities, whether they be chemical, biological, or therapeutic, available to us. Let's bring this capability, this generative capability, to life through an example. What I'm showing you here is an example of one protein, where in green I've isolated the functional part of that protein that is responsible for its function, binding to calcium in our cells. We can take that part and isolate it and use this as a kind of prompt to our model EvoDiff, which can then take that prompt and learn from the information that it's been trained on and see to now design a brand new protein sequence around that part, step by step, from the bottom up, creating a brand new protein sequence that's never existed before. And what's amazing is that this new protein, designed by our AI model, shows a structure that we can then explicitly link to biological function measured in the lab in the real world. Now, I'd be remiss to say that it stops there at our ability to generate new molecules and proteins. That's just the start. To actually learn and understand this language of biology, we need to be able to reason across scales, across different types of data, and put all these components together, from the molecule level all the way up to that of the patient. We see this as a foundation by which we can now close the loop and generate a data and learning flywheel to close this vision of precision medicine for once and for all, to be able to personalize discovery, experimental design, and ultimately, hopefully, realize more effective treatments. Thank you.